Good evening, everybody. Welcome to BrisScience, the University of Queensland's free public lecture series on science, where we aim to bring you not just the best communicators, but not just the best scientists, but also the best communicators, as you will see tonight. We are proudly supported, of course, by the University of Queensland and hosted here at The Edge, our wonderful venue partner. I'd also like to respectfully acknowledge the traditional owners of the land on which we're meeting tonight and pay my respect to elders both past and present. And I'm your MC for this evening, Joel Gilmore. Thank you all so much for coming out. I think it's going to be a great evening. Special shout out to our guests from Rochdale State School up the back. Always great to see our students out. So a couple of bits of housekeeping first. If this is your first BrisScience, welcome, hello. We do a couple of things a little bit differently here. We're going to have our speaker come up and they're going to talk to us about speed breeding of plants tonight. And then we are going to take questions. You can do that in two ways. You can either do it over Twitter using hashtag BrisScience. Sometimes it's right there on the screen behind me, it's perfect timing. But hashtag BrisScience, or you can ask questions on the pieces of paper you picked up on the way in, or anything. If you write them down on a $20 note, we'll give them special treatment. Take those questions, and at the end of the talk, Dom or Leonie will come around, collect those questions, and we'll get through as many of them as we can. If you do have your phone out, though, could you put it onto silent, please, just to make sure we don't get interrupted. And then afterwards, we will have some food and drink and a chance to talk with Lee a little bit further. Otherwise, let's get onto it. So tonight, I'm very excited to welcome Dr Lee Hickey from the University of Queensland, who is a geneticist with the the Queensland Alliance for Agriculture and Food Innovation. Lee investigates the diseases of crops, such as wheat and barley, and the genetics, and how we might be able to improve those plants. And one of his big achievements, one of many, is a way of speeding up the breeding cycle of plants. And it was for this, amongst other things, that he was named the 2017 Queensland Young Tall Poppy Scientist of the Year, although perhaps tall wheat stalk might have been more appropriate. Could you please join me in welcoming Dr. Lee Hickey?
Thanks, Joel. Thanks very much. Good evening, everyone. Thanks a lot for coming. And look, I'm going to give a talk tonight about speed breeding, not speed dating. So if there's any confusion, please leave now. Something that's very, very different. But look, I hope that tonight you're going to learn something new. About plant breeding or genetics or agriculture in general, and hopefully maybe if I get lucky I'll blow your mind just once, just a little bit, so get excited. First of all, I want to transport you to a very familiar place, Coles, Woolies, Aldi. I'm an Aldi guy actually, but I want to ask you a question. How often do you, when you're shopping, think about where the food comes from when you're in the shopping centre, in the grocery store? I have to admit that when I'm shopping, I'm looking at the brands. I'm also conscious about my budget. I'm looking at the prices. Often, admittedly, I'm choosing a cheapest product in many cases. And so, the thing is, our food comes from the farm. And, it's even like agriculture is a bit of a dirty word for people in the city, But that's actually true because there's dirt on farms, and so farms are really important. And I have to it's probably fair to say that many people in the city are not farmers. Farmers exist in our rural communities, unless of course you have a veggie patch in the backyard. And does anyone have a veggie patch in the backyard? Wow. Oh, so many hands, especially from Rochdale up there, back there. Very good. Because when I was a kid, I had my own veggie patch. I was a bit of a strange kid, I suppose. I would actually rush home from school to tend my veggie patch. And and I had all the sowing guides and, when to grow the different vegetables. And you know what? I was fantastic at growing all the vegetables that I really hated to eat. It's really bizarre, but I love just growing them. And I would grow chocos, and they'd be going crazy, and I'd actually be giving them away to my neighbors, and they're refusing. Oh, no more chocos, Lee. We've got enough, but I haven't given you any yet. So anyway, look, I've got some vegetables here, and so some of the delicious vegetables, very healthy, good for you. Broccoli. Are there any fans of broccoli in the crowd? Okay, this is where it gets exciting. So, broccoli fan over there. Oh, intercept. If only you were on the Queensland team. Another broccoli fan. Oh, we might go for right up the back there. I was aiming for you. Oh, wow, it's like a football, really, or soccer ball. The World Cup's coming up soon. How far is Australia going to go? Jeez. And a little one. I'm sure I can throw this a little bit further. Maybe over here in the corner. Oh, beautiful catch. And kale. Any hipsters in the room? I can make fun of hipsters because I'm a hipster, and that's the way it works. Here we go. Some kale down here. Oh, good catch. Oh, and one over here. Excellent. Good work. And Brussels sprouts. Who doesn't like a Brussels sprout with a little melted butter on top? Oh, just delicious. I'm going to go for Rochdale down the back. Oh, no. Incoming. You get a Brussels sprout. You get a Brussels sprout. Everyone gets a Brussels sprout. Paying attention? I feel like Oprah here. No, there's no cars under your seat, so just calm down, everyone. So, I'm going to let you in on a little secret. All these vegetables I've been dishing out to you all. Actually, I want them back after the show, please. No, it's fine. You can eat them. And I haven't added any weird genes to them. They're perfectly safe. They might have a bit of dirt on them. But I'm going to let you in on a bit of secret. A little secret here. All of these plants, they all come from the same plant, the wild mustard plant. And so humans actually bred and selected and developed these different vegetables that we have today in the grocery store. There's no such thing as a wild broccoli growing in the forest. Humans created them. Humans selected different traits derived from the wild mustard plant to create our beloved kale by selecting for leaves or selecting for buds to create Brussels sprouts. Mind blow, right? Now I dare to challenge, what is a genetically modified organism? That's pretty modified, and they don't exist in the wild, and we created them. That's pretty cool. Humans are cool. So keep that in mind for the rest of my talk, please. Now, on a more serious note, and it might be a bit of a myth that, hunger, we've got enough food in the world. Hunger is a thing of the past. And certainly living here in the city, we do have a plentiful food supply. We just have to go to Woolworths and Coles. But in reality is that in many developing countries, hunger is still a big problem. And in fact, globally, hunger is still the number one cause of deaths. And we weigh it up against some of the other disease-related deaths, like heart disease or HIV, and really outweighs this remarkably. In terms of the future and our food supply, there's a lot of factors that are currently threatening global food security. A lot of countries around the world, we have a lot of food at the moment, but it doesn't take much to upset the whole supply. And a good example is a drought in Russia in 2010. Dramatically, we saw a rise in prices of global wheat price. What it means is that if a couple of key events come together simultaneously, a flood in India or a drought in Russia or something else on another continent, then we can see chaos come into play. And that's particularly important in a lot of regions that actually have unstable governments. So, but to list them off, well, of course, we have climate change right at play, fertilizer dependence, emerging diseases, so new diseases that we didn't normally have to deal with. And, of course, the current diseases that the farmers have to deal with are rapidly evolving as well. So there's a constant battle against these things. And I would have to say, I'm going to add it there, is that the consumer acceptance of GM crops is also important, particularly for developing countries. Now, crops like wheat, I work a lot on wheat, so you're going to hear a lot about wheat tonight. And wheat is really an ancient crop. In fact, hunters and gatherers were collecting wild emmer wheats, the most productive ones, keeping them, selecting them, planting them. And so they were the first plant breeders, right? Today, we have people employed to breed these crops. But if we transport back in time to ancient Rome times, they even had a god who was responsible for causing disease on wheat crops, in particular rust. And so at the beginning of the farming season, they would actually make a sacrifice to please the god. And that usually, unfortunately, involved slaughtering a young puppy and sprinkling the blood somewhere. I don't know. But that was crop protection in Roman times. We fast forward to today, and we're not making sacrifices. We have science and technology. And one of the tools that farmers have today, of course, to protect our crops, make them more productive, stand up against all these diseases rapidly evolving, is fungicides and pesticides to control insects. These chemical tools have really boosted productivity of modern agriculture and have supported this exponential growth of humans around the planet. Of course, nobody likes their food, the idea of our food being sprayed with chemicals. So I guess the whole community is really trying to drive forward in terms of eliminating these chemical inputs or reducing them as much as possible. And that's where new science and technology comes into play. But the other way that we can control disease in our crops is through harnessing genetics. A fun fact, the wheat genome is 5 times bigger than the human genome. That's why they're so smart, the wheat plants. But that's something like, I think it's 15 billion base pairs. I wonder wheat breeders have it hard. But I'd like to mention Sir Roland Biffin from Cambridge. He was the first one back in the early 1900s. He took a leaf out of Charles Darwin's book and he applied Mendelian genetics to wheat for the first time to discover that resistance to disease was under simple genetic control. And so, here you have here on the slide a nice resistant wheat leaf and a susceptible one, overcome by rust disease, where you originally have to slaughter a little puppy to please the gods. We actually have genetics which controls this feature in our plants. And so, what it means is we can actually deploy resistance genes in the plant, in the crop variety, and the farmer doesn't have to spray their crops with fungicides anymore. So this is what modern plant breeding is all about. But the problem is that these diseases, these fungal pathogens, are so shifty creatures. They're constantly mutating and overcoming the resistance genes deployed in our crop varieties. And so this is an ongoing battle. And if we look here, this is just to give an example of the migration routes of wheat stripe rust moving as the pathotypes change and mutate on those resistances. They're actually moving in jet streams. The spores are so small that they can travel continents. They can travel from the UK to Australia, or India to Australia, or the US to Australia. And it only takes one spore to land on our wheat crops and multiply and destroy our whole wheat industry. Not only are the existing diseases are rapidly evolving, but there's new diseases popping up that we're not used to. Wheat blast was actually first detected in South America. And it actually evolved from being a disease on rice, so which is pretty crazy. And it actually appeared for the first time in 2016 in Bangladesh, a region where a lot of people depend on wheat production to feed people. And all the varieties were susceptible. And so, how long does it take to replace those varieties in farmers' fields with a resistant one? The wheat blast is so devastating, it just destroys the spike completely, and no grains are actually produced by the crop when circumstances are suitable for disease development. Climate change, another big factor that imposes a major challenge for us producing food here on Earth. Here's the current snapshot of Australia and the chance of above median rainfall over the next 2 months for our wheat farmers in Australia. And you can see it's not good. It's quite a low chance. Most farmers have sown seed and they haven't got enough rain to get the crop up, or the seeds germinated just enough to get going, but now it's starving of moisture and dying. So it's not a good time and at the moment for our farmers, but looking forward into the future, these sort of variable weather patterns are more likely to occur. Temperature is also a big challenge. The current studies out there that are projecting crop production under future scenarios are spelling bad news for our farmers. So for every 1 degree temperature increase, we're talking about losses in the magnitude of 6% for wheat yields globally. So wheat doesn't like warm temperatures at all. So through plant breeding, we can certainly deploy traits which help the crop use water in a smart way. A good example of this, and we're doing a lot of research on this topic at UQ, but where we can actually deploy traits like deep roots, where the crop can actually access stored soil moisture deep in the soil profile and avoid a drought or minimize the effects of drought. So will we meet the future demand? Well, the countdown's on. 2050, that's what everyone's counting down to. And we're talking about feeding 10 billion people or so. Who knows what the exact number is going to be, but it's a hell of a lot of people. And this figure sums up nicely, I think, about where we're headed and what we need to do in terms of adopting new science and technology. This is the global trend for wheat yields in time, where the yellow line is our current trend. The green line is actually where we need to be to produce enough food. So we're talking about producing an extra 60 to 80% food globally to feed everybody. Now, the red line is where we could end up. All right, if we stop plant breeding today, our crops fall vulnerable to climate change, these rapidly evolving diseases, new diseases. This is where we end up, a shortfall. And if this is happening for all of our major food crops, we're going to be in trouble. Now, unfortunately, we're on track to do much better than that. But what we need to do is we need to lift our game, and we need to go much, much faster. And so we can only do that through improved genetics, accelerating the breeding process of these crops, and deploying better agronomy practices, so management practices on farm to close their yield gap. So one of the key problems is the whole rate of improvement of our crops. Because if we had better crops tomorrow that were climate resilient, disease resistant, then it wouldn't be a problem. And one of the problems is that developing improved crop varieties is such a slow, long, and boring process, unfortunately. So this is a bit of a figure of a summary of a wheat breeding pipeline. So in a nutshell, without showing all the details, these are the 3 main phases of a wheat breeding program, okay? We have crossing and inbreeding, and during this phase, the plant breeder is making crosses, generating inbreds, which I'll explain shortly, which are needed to generate genetically stable materials that undergo extensive testing, and for yield, disease, grain quality, how good the varieties are making bread or noodles, right? And that takes 4 to 5 years. And then you enter a seed increase and release phase, and that's needed to bulk up the seed of the new variety and sell it and make it available to our farmers. Now, the back end of this program is pretty fixed because we need that testing to make sure that the variety is going to stand up in the paddock and that it's going to be reliable for our farmers. But it's the crossing and inbreeding phase where there's a lot of room for improvement, generating better genetics entering this second or back end of the breeding program. And mind you, this whole process can take up to, in pretty bad circumstances, it can take 20 years for some crops. But, the breeding companies in Australia are doing a bit better job, going a bit faster, and so getting that down to around 8 years is quite common in Australia. But if we focus in here on the crossing and inbreeding phase, I can walk you through what would be a traditional inbreeding program. So the plant breeder making the crosses in year 1, so taking the best plants, the disease-resistant ones, the drought-tolerant ones, the high-yielding ones, and crossing them, mixing up all the genes. And plant breeding is a numbers game, all right? There's lots and lots of genes they have to combine. And then in the second year, because if you're only growing one generation a year, this is what you're doing. You grow the F1, which are the hybrids, right? The first generation. And then the F2 is where everything segregates. It's like, anyone heard of Mendel's peas? This is where you do your Mendel's peas experiment. But, for the inbreeding species like wheat, barley, rice, you continue on because you want genetically stable or inbred plants from that cross. Because then you can replicate testing in the different environments and so forth. And that requires a number of generations of selfing or inbreeding to generate what we call F6 lines. And these are about 96% inbred. And plant breeders say, that's enough. We'll call them inbred for now, because you can't actually get completely 100% inbred plants. There's still residual heterozygosity. Now, keep that in mind. Plant breeders in Australia are going a little bit faster, so they might actually grow an extra generation a year in the glasshouse, because over summer it's too hot outside, but in an air conditioning glasshouse you can do an extra generation. And if you do 2 generations a year, look what happens. You can do it in half the time. Easy sums, right? Now, this brings me to rapid cycling wheat. Back in the early 2000s, my colleagues were really inspired by NASA growing wheat in space. They were trying to anyway. And they were teaming up with Utah State University in the US to do this. It reminds you, does it remind you a bit like a movie, growing potatoes on Mars maybe? Although this story doesn't involve Matt Damon, I'm sorry. And so, NASA were trying to grow wheat in space. And how were they doing it? Well, they were exposing the plants to continuous light, so the plants didn't sleep. So it's a complete myth. The plants don't need sleep. And what this did is it tricked them into the reproductive phase, flowering, and producing grain much, much faster than normal. And so we thought, what a cool tool this could be to speed up our crop improvement here on planet Earth, right? Our research, our breeding, everything depends on how long it takes to grow the plants. We're just sitting around waiting for our plants to grow. So if we can speed it up, this would be a pretty cool tool. And so, look, incidentally, Utah State University developed this variety, very famous, apogee, very short. You can see half of it spike, all grain. But they needed to do that to grow a very short variety in space, because there's not much space in space. And incidentally, just recently, they just released some footage of wheat growing on the International Space Station. So all these years later. And so hopefully we can get the video to work. Here we go. YouTube never fails. And so what you're looking at here is on the International Space Station, the size of a small beer fridge, and wheat growing on an artificial gravity system and rapid cycling, seed from seed, very quickly. And they're using these environmental conditions to accelerate the plant development, trick the plant into flowering very early. And so this footage was only released probably a month ago or so, which is pretty amazing. So they succeeded. And meanwhile, back on Earth, we started trialing this system back in the early 2000s at UQ, and we called it the speed breeding system. Speed breeding, right? Not speed dating, different. And so when we first started doing this, a lot of skeptical people out there. People told us, you can't do that. That won't work. Because that was pretty extreme, right? NASA were trying to grow wheat on space, and they were using this crazy system of exposing the plants to continuous light. That won't work. And look, in fact, when we first tried, fair enough, the plants were pretty runty and pretty pathetic, but that's because this was the first time that we actually tried to grow wheat. But once we optimized the nutrients and the temperatures, the plants look great. And they even resemble the ones in the field. And people that come to visit are really amazed at how good quality the plants are, but they're growing super fast. So now we use this speed breeding system daily to accelerate all of our breeding and research at UQ for wheat and barley. Now, incidentally, over the last 12 to 18 months, there's the urgency, it seems, is growing to speed up the development of our crops. And I've noticed this because I've received overwhelming requests from scientists and plant breeders from around the world to asking how do we grow our plants quickly? How do we set up a speed breeding system at our institute and our breeding program? And so I guess we also were not the only ones who were speed breeding by this time. There are other groups around the world who caught on, developed their own systems, and so we decided to team up with a couple of those other groups, namely the John Innes Centre and the University of Sydney. The John Innes Centre in the UK, one of the biggest plant science centres over there. And we decided to make our protocols publicly available so everybody had the information, how to actually grow these plants quickly, because there wasn't a lot of information out there. And let's face it, the communication was really terrible. You'd be amazed at how many places I went when people didn't even know or heard about speed breeding. A guy came up to me at a conference and said, how do you grow 6 generations a year? And I said, mate, just leave the lights on. A little more to it than that, but it was half joking. And so fortunately, our protocols were published in a very fancy science journal this year on the cover of Nature Plants, and which was great for exposure for the technology and spreading the word. But before I go on, I need to thank these key people involved. A lot of people, but the co-lead authors, Amy Watson, my PhD student at UQ, Shreya Ghosh, PhD student at the John Innes Centre under Brando Wolf. And Brando Wolf played a key role in setting up speed breeding there in the UK at the John Innes Centre. So in a nutshell, speed breeding, we can achieve 6 generations of wheat per year. And this is how we do it. We leave the lights on, literally, for 22 hours. And we're nice to them. We give them 2 hours of dark to rest. And during that dark phase, we lower the temperature from 22 during the daylight hours to 17 degrees. And we found that extra 2 hours of rest, just like humans, we need our rest. It seems to improve the quality of the plants, but doesn't really compromise too much on speed. So this is pretty good. Plus, you save a little bit on power because you don't have to run the lights an extra 2 hours. But this is in comparison to a regular glasshouse conditions where we can really only achieve 2, sometimes 3 generations a year for some wheats that are a bit faster. So it becomes quite a powerful tool for crop improvement. And incidentally, we tested a bunch of other species and it works too. So barley, chickpea, we can also achieve 6 generations a year. This is in comparison. We can only get 1 in the field, of course. We grow wheat in the wintertime. So we sow normally. Our farmers sow around May, sometimes they're getting a bit earlier, late April, and they harvest around November time. And in summer it's too hot. Incidentally, canola, it works too. 4 generations instead of 2. It takes a bit longer because the seeds are a bit oily and they need a bit longer to form before we can harvest viable seed. Now, I know what you're thinking. 6 generations, Lee, that's a lot. And I don't know whether this is magic or science. And that's exactly why I have the next slide, because seeing is believing. And here we have a beautiful time lapse from the Earlham Institute in the UK, another one of our collaborators there on this paper. And we decided to capture the rate of the growth. Speed breeding on the left, normal breeding on the right. It's a race. And speed breeding plants off to a great start, already elongating. Here are the stems, here's the flag leaf, very important. The spike has emerged and flowering, and our speed breeding plants are just starting to elongate now over here, while our speed breeding plants are already mature, and we can't wait that long. We have to cut it short, I'm afraid. Normal breeding is just way too slow. And here is some really beautiful pictures that my fabulous PhD student Amy Watson captured from some experiments where we grew these plants because we actually needed data to publish these protocols. We were using these protocols together with plant breeding programs, but in order to make this information available, we had to go back and get the data and show how effective this system was. So we grew all these experiments, and here you can see the plants sown on the same day in 2 different glass houses side by side, one fitted with speed breeding lights, 22 hours, the other one just natural light, about 12 hours each day. And here on the left is speed breeding versus regular glasshouse on the right, captured at the same time point. So for wheat, barley, chickpea, canola, and it even works for a bunch of model plants, which are very important for the plant science community. So like Brachypodium is a model for cereal crops. Brachypodium and Medicago, it also works. And incidentally, it also works for peanuts. And peanuts, it was a bit of a myth out there where, peanuts were considered to be a short day species, and so it wouldn't work for peanuts. But, some scientists, my colleagues at UQ, decided to just give it a go. And what do you know? It worked. It worked for peanuts. And so the peanut breeding company in Australia actually cycle all their breeding materials now through speed breeding to get 3 generations a year instead of 2. And it's probably the most impactful thing that you can do in a breeding program when you're only doing 2 generations a year. And so the list continues, and it's sort of growing daily, what species are compatible. On Twitter, I found out a really good plant scientist over there in Germany, Carl Schmidt. He has produced a really nice speed breeding protocols for amaranth and quinoa. All the hipsters in the room, quinoa, you can speed breed quinoa. And actually, the amaranth is a really interesting story because it's a short day species. So what that means is that shorter days trigger reproduction phase or flowering. Okay, so if we leave the lights on for 22 hours a day, it shouldn't trick them into flowering. They need short days to induce this flowering. And so what they did is they come up with a really cool recipe, and this highlights how flexible speed breeding is that they actually grew the plants under continuous light during the vegetative phase and then switched to short days for just a few days, and it triggered the flowering and then continued on continuous light. And by doing this pretty cool, innovative recipe, they can achieve up to eight generations a year of amaranth. And so I think that is a perfect example why we can speed breed perhaps just about any species. You just got to find the right recipe. Lentils. This is a nice example where we're collaborating with the University of Melbourne. This little lentil on the right, it's actually a landrace. The one on the left is actually an Australian variety grown by our farmers. But the one on the right has good genes for disease resistance and drought tolerance. But the problem is, under regular conditions, it takes 160 to 180 days to flower. Under speed breeding, 27 days. So this tool is opening up a whole new gene pool that we can use to improve our modern crops. And like I said, the speed breeding recipe is really highly flexible. It's highlighted by our different systems that we have across our institutes. For example, we use a temperature-controlled glasshouse at UQ fitted with supplemental lighting. This is our first generation speed breeding system. The first generation speed breeding system at the John Innes Centre used quite expensive Conviron growth chambers where they do a lot of their research. And at the University of Sydney, they adopted this pretty innovative, low-cost growth rooms, just in a regular room, fitted with an office air conditioner and LEDs. And they're actually accelerating a lot of their research, achieving a lot more generations per year. Now, speed breeding 2.0, other than just having a really cool name, is actually really important. And it's all about reducing the operating costs of this technology. So like all technologies, they're always really expensive when they first get developed. Like DNA markers, now they're super cheap. What we want to do with speed breeding is really reduce the costs, and one of the key innovations there is in the lighting, of course. And so we've recently replaced all of our lighting from our old sodium vapor lights to these new generation LEDs, which are only emitting the wavelengths that the plants need for healthy plant growth. So that's only the blue and the red. And so no other wavelengths are needed. And this means reducing energy for the lighting, right? But also the heat load is reduced. And this means reducing costs associated with the air conditioning in those growing environments. And that's pretty high cost in an environment like Australia, where we have pretty warm conditions all year round. I love this picture. Beautiful picture of the speed breeding facility scaled up at the John Innes Centre. And that's because all the plant scientists there now are using speed breeding. Originally, when they first trialed the system in their little chambers, a lot of people were very skeptical. But now they're fully convinced, and everyone is using, in the whole centre, speed breeding. So they needed to adapt speed breeding to large facilities. And I just love the side of this. The purple coloration from the glasshouse. They even have a picture where the glasshouse is covered in snow, really highlighting what speed breeding's doing out of season, rapid cycling our crops to develop better crops for our farmers. So if we look back at our breeding pipeline and where exactly speed breeding fits in, well, it's in that original phase, like I mentioned. It's about shrinking this crossing and inbreeding phase and getting that cycle time down with huge potential to increase the rate of development of our varieties for our farmers. A lot of my team is using speed breeding, and over the years we've developed methodology for actually identifying disease-resistant plants in the speed breeding system. Because if we can grow plants quickly all year round, and if we can develop a tool where we can actually identify the plants that are resistant, this is pretty advantageous, right? Because what it means is we can select the resistant ones, and we can only grow them in the field. We don't have to grow susceptible ones in the field. And so we can make a lot of gain with these sort of approaches. Very simple. We just infect the plants as they grow, and we select the resistant ones. And so we've developed really nice methodology for doing this for some of our major diseases that affect wheat in Australia and around the world. Yellow spot or tan spot, my PhD student Eric Dinglerson, he's actually developed a nice system for this. It's actually the #1 disease of wheat crops in Australia. Most of our varieties are susceptible, unfortunately. So we're hopeful of fast-tracking resistant varieties. Adnan, he developed some nice systems for resistant to rust diseases, I mentioned earlier, but we have leaf rust, stripe rust, stem rust, they all attack wheat. And Fusarium head blight is a devastating disease, again mentioned earlier, similar to the blight disease that I mentioned earlier in Bangladesh, but you can screen for this under speed breeding as well. But what it means is we can screen all year round, no matter what the weather conditions are, and they can be tricky sometimes, because normally the traditional way of doing this is in the field, right? And so it's a pretty useful tool. Now, to highlight the power of speed breeding and disease trait selection, we actually did a case study, and we picked this variety called H45. It was really popular by farmers, particularly in New South Wales and Victoria. They loved to grow it. It was high yield potential, and they just knew how to manage it really well. But unfortunately, what happened is a new strain of rust emerged and rendered that variety completely susceptible. So susceptible that fungicides were no good. And so we picked this variety, and we thought, okay, let's transfer disease resistance into this variety and resurrect H45 for our farmers. Let's make it resistant with a whole stack of resistance genes. So we performed a rapid back-crossing strategy in the speed breeding system, repeated cycles of back-crossing and selection for the rust resistance, and, well, this is the end product. And 18 months later, we had to perform a seed increase to actually generate enough seed in the field, and that slows you down. That takes 1 year. But once they entered the yield trials, this is a photo actually taken at a farmer's field day, attended by more than 300 growers in Victoria. and they couldn't wait to get their hands on this wheat variety because it looked like H45, it smelled like H45, but it had the rust resistance, right? So, I mean, this photo kind of reminds me of when you go fishing. You know, you're very proud, you got your fish. But look, I was proud. When you develop something like this in plant breeding and in science, it's useful, you know, it's pretty cool. Another great example is the first variety. In fact, we just released, in partnership with Dow AgroSciences, completely bred and developed under speed breeding. And its name is DS Faraday. It represents Australia's first high-protein milling wheat with tolerance to pre-harvest sprouting, a problem that people in Australia, researchers, farmers, have been trying to solve ever since we started growing wheat in Australia. And all of our varieties are completely susceptible. So pre-harvest sprouting occurs when you have a mature crop of wheat and unfortunately it rains or there's a storm and all the seeds germinate and sprout. Pre-harvest sprouting. And that makes the grain terrible for making bread. You can't make bread out of it, and certainly people don't even want to feed it to animals because fungi colonize it, all sorts of problems. 1 in 4 or 5 years, this is a massive problem. And so we developed the first variety with a step change in the level of tolerance to pre-harvest sprouting using this speed breeding tool. And how do we do it? Well, very similar approach to our H45 variety. We did these rapid cycles of backcrossing and trait selection. We actually selected for grains that didn't germinate at harvest time. We break the dormancy using a cold treatment and then grow them on in the speed breeding system and just keep doing that. And we transferred about 10 or 12 genes for grain dormancy. We have no idea where they are in the genome. That's way too difficult. As I mentioned, 15 billion base pairs. But it doesn't matter. We did it on phenotype. We did it on the trait itself. And of course, the dormancy wears away 3 or 4 weeks after harvest, so the farmer can grow that variety next season and it germinates no problem. Another really cool tool, or application rather, for speed breeding is tapping into our gene banks, right? We have all this massive genetic diversity just locked away in storerooms, basically. You know, well, one day we'll use that. We haven't done a very good job at using that genetic diversity to improve our crops, our modern crops. And that's because it just takes so long to transfer the traits, the disease resistance, the drought tolerance, in these very old land races or wild relatives that are in the seed stores, transfer those traits into modern varieties. It takes many, many cycles of backcrossing, and we need to separate all the deleterious genes. There might be deleterious genes for grain quality, or they might be too tall. And so with something like speed breeding, it opens the door. It's a game changer. We can bring in this diversity and do it quickly in 1 year. 18 months, right? It doesn't have to take 20 years now. And so my team, one of the collections we're working on, mining for new traits, is this Vavilov wheat collection. Vavilov was a very famous Russian botanist, and he traveled the world collecting seeds and tubers. And this was before modern plant breeding, back in the early 1900s. And he collected all these diverse specimens and brought them back to a seed bank in Russia, where they're all preserved today. And so we're working on some of these land races, that even some that Vavilov collected himself, to actually transfer these traits quickly into modern varieties, particularly with a focus on disease resistance. I heard someone say, can you combine speed breeding with other technologies? Yeah, of course you can, and this is where it gets exciting. Okay? Now, don't panic. I'm going to talk about GMOs. Now, I have to say that, I don't actually do much work on GMOs or GMO technology myself. I'm more using the speed breeding system to manipulate the genetics very quickly and through crossbreeding. But we do use GM to validate some of our research, validate some of the genes. And that's because, unfortunately, GM technology is not accepted in wheat and barley. There's not one single wheat variety that's GM that's grown in a farmer's field around the world. But for some crops, we're quite happy to grow GM crops, right? Cotton is almost 100% GM in Australia, and we wouldn't have a cotton industry without the GM traits. Canola, there's quite a lot of canola that's GM grown in Australia. So in short, we're happy to wear the cotton underpants that are made from a GM cotton plant, but we're not happy enough to eat the wheat from a GM cotton wheat plant. But GM technology can really create opportunities for disease-resistant crops in shorter period of time, and even healthier foods. And I think that's where this tool can really be useful. A great example is the golden rice, particularly developed in regions in Asia, where a child in Bangladesh can get 60% of the vitamin A requirement from just 50 grams of golden rice, right, each day, which is pretty remarkable. It's not 100%, but why deny this sort of technology that's going to really improve the livelihoods and health of people in these countries? Another example is blight-resistant potatoes. They have a genetically modified blight-resistant potato, all right? And this is a disease that caused widespread famine in Europe. Even locally, we have some really neat work going on here in Queensland, QUT, where they've actually got GM-resistant bananas that are resistant to the tropical race 4 strain of Fusarium. We're going pretty close to not having bananas in the future here in Australia, and I would be more than happy to eat one of these genetically modified bananas if it means that our farmers can grow bananas in a sustainable way. So GM technology has its place, I think, and it's a pretty useful tool from a plant breeding perspective. And so where does speed breeding fit into all of this? Well, it can certainly boost these transgenic and genome editing pipelines. Any pipeline where we have to go through the lab and go through this tissue culture process to edit the gene or insert the gene for disease resistance, say, we need to regenerate the plants. And this is pretty slow. Again, scientists just sitting around waiting for our plants to grow. And so with this sort of approach, we're talking about saving 3 months from this transgenic pipelines. And what this means is new traits into farmers' fields faster, and hopefully the new technology, CRISPR or gene editing, will be accepted for improving our crops for our farmers. Now, here we're going to dive into a bit more science, so hold on. But to make it easier to understand, we're going to use bacon as the example. Genomic selection was actually developed back in 2001, or at least the theory. And it was actually developed by one of our colleagues at UQ. He's just recently come back to Queensland. And he actually was one of the brains behind this genomic selection concept. It was originally applied and developed in animals. And so in this example, we're going to use pigs and bacon. But essentially, here we have a population of pigs, and we have known genotypes. We can genotype them because DNA markers are cheap now. And we have their phenotypes. We know how much bacon they produced. Don't ask how. And so we can generate a really nice prediction equation pairing this information, the phenotypes, or bacon, with the genotypes, or DNA markers, or barcodes, if you like. Every barcode or genome is unique. Every pig has a different one. Now, we might have a selection candidate. So this might be our breeding population, where we're actually trying to improve bacon production. And what we can do is we can take those pigs, and instead of test them for bacon production, we can actually genotype them, because it's cheaper than actually phenotyping them, and we can predict how much bacon that those pigs would produce. And we can do this at any stage just by genotyping a very young pig, for instance. And so this is all running on these very sophisticated prediction algorithms, right? A lot of mathematics involved. So that's how it's applied in animals. And certainly now we have Ben Hayes, who was one of the brains behind this approach in animals. Now he's working at UQ. We're now applying these strategies in crops, and we're pretty excited about the opportunity. And here's just a visual representation again of a crop breeding program where we have the crossing going on, developing inbreds, and then we would test them once they're genetically stable. They go into the paddock, we test them for yield, quality, and then go on to variety release, and we'll pick the best ones, and they'll keep on cycling like this, like a big washing machine. Now, genomic selection, instead of doing all this testing, because it's a long time and very expensive, actually costs $50 for one single plot like this. It costs $50 in the ground. Crazy. We can genotype a wheat much cheaper. And so we can actually predict, using these algorithms, the best individuals and shortcut this whole breeding cycle. But look what happens when you introduce speed breeding into the equation. We can start to cycle these things even faster, which is pretty exciting. And while we're doing this in practice with breeding companies here in Australia to actually try out these new breeding approaches, we couldn't wait, and we had to do a simulation study just to see how effective this could be. And so we actually did the simulation study using real wheat data sets from the Global Wheat Breeding Program at CIMMYT, it's based in Mexico. And we tested the traditional breeding approach versus what we call a speed GS approach. You've got to have a cool name these days, where you actually combine speed breeding, so you're going fast generations, and you're applying genomic prediction or genomic selection, so you're shortcutting the testing phase. And if we come up with a really cool strategy like this, we can look at the gain we can achieve. After 30 years of breeding, we end up with something like 34% genetic gain, extra, right? And Kai, my colleague, he's in the crowd, very tall-looking Viking guy over there. He did the sums, and he worked out that that's more than 1,000 loaves of bread per hectare. We talk about feeding the world. This is the sort of innovation and a different approach that we need to use to speed up the development of our crops. So lastly, to wrap up, some final thoughts from me, and I guess we'll have an opportunity to have a bit more of a discussion. Plant breeding companies in Australia and around the world already adopted speed breeding. If they're not, they need to invest in facilities because it's quite expensive, or they need to outsource it as a service. One of the things I'm quite excited and motivated to work towards is taking speed breeding off the grid. There's booming technology when it comes to solar panels, lighting. We're excited about LEDs. LEDs are old, man. We have laser lights now, right? Anything to do with lasers are cool, but lasers can actually be outside of the growing environment and beam in and hit a diffuser and diffuse a light across the plants, removing the heat load completely out of the growing environment. We have glass now, sheeting, that doubles as solar panels and lets some wavelengths through for plant growth while capturing other wavelengths. So this technology is rapidly evolving and rapidly developing. I believe that we need to use all the tools in the shed to meet the 2050 challenge, and that includes GMOs. We need to be resourceful, and we need to be adopting this new technology and science and embracing this. If we stand a chance to make our agricultural systems productive and sustainable and profitable for our farmers in Australia. I pose the question, why can't GMOs or GMO crops coexist with organic farming? To me, it just makes sense. We're all trying to achieve the same goal. We're trying to eliminate chemical inputs in producing our food, right? That's what all the researchers around the world, like myself and my colleagues at UQ and other institutes, we're trying to develop technology that achieves that goal. So I think there's got to be compromise, right, on both sides. But why can't we have a GM organic farming system? Think about it. And this is a really exciting opportunity, I think. And because there's a massive trend in people trying to grow more food in cities. And that's because people like locally grown food, right? And it's pretty interesting, particularly around the indoor farming and vertical farming scene. There's even Elon Musk's brother. What's his name? Kimball Musk. He's actually opened a venture in Brooklyn where these are actually modified shipping containers, decked out with LED lighting, growing plants like a vertical library. Really amazing. They're actually growing local greens and serving a local market in Brooklyn. And I reckon that speed breeding could help, or a speed breeding approach could help boost productivity in this system. Can we take other species that are traditionally grown outdoors and grow them indoors more productively, more cost effectively? It might be just a matter of tweaking a couple of key genes. And with that, I'd like to acknowledge all the people involved in the speed breeding paper, particularly from around the world, the different institutes in Australia and overseas. Lots of people collecting all these big data sets to put together, make all of our protocols publicly available and spread the word. And of course, our funding bodies, the ARC, very important here in Australia, the federal government, and the GRDC, which is actually indirectly, these funds are coming from our farmers. And a funny picture of my crew. Thanks very much.
Thank you so much, Lee. We'll give you a chance to have a drink. We will take questions now. So if you've got burning questions, either time to boot up Twitter or wave them around if you've got them written down, and we will come around and collect those. Meanwhile, obviously this is one of the many outreach activities that UQ is involved with, so check out the UQ website for some of the other great science talks we've got going on. We've got JFO coming up, a great camp for physics students particularly, and more things coming up like that, so hop on the website and check those out. Next month, we are, of course, back with BrisScience, and we are talking about mummies. Not the family kind, but the ancient Egyptian mummies. And so, thank you so much. We're going to look at how mummies were created, the hows and whys, natural mummies, artificial mummies, and everything else that goes along with it. So make sure you don't miss that. Lee, you ready for some questions? Okay. Questions. So, first question is, actually I might get you to take centre stage so that you're unencumbered there. Did plants that were developed via speed breeding perform well on the farm? So did it translate okay from the lab to the farm?
Yeah, this is quite a common question. Essentially, we're using the speed breeding as a tool to assemble the better genetics, develop those inbred plants, and put them in the field. Of course, we can't replace the field testing. I sort of highlighted that in my talk. The field testing component, we still need to test those inbreds across many environments where the farmers are growing these crops to make sure that they're performing, and that's normally done over 3 or 4 years. But before we do all those extensive trials, we actually have to bulk up the seed because we only get a small amount of seed from the speed breeding system. So typically, all of these plants come out of the speed breeding system and we sow the seeds in a seed increase trial, and that's just a 1 or 2 metre row. They produce lots of seed, 1 or 2 kilograms of seed, and that's plentiful for sowing across many, many environments and doing this thorough testing. And when we do that, we actually remove these maternal effects that we get from the speed breeding system. We get high quality seed going into that trialling system.
Great. Next question is, how long will it take for these innovations to make it to my dinner table?
Well,
no pressure on your future funding or anything.
What's the standard answer is normally 20 years or well, look, we've just released the first variety completely bred and developed in this system in partnership with one of the companies. So farmers are starting to grow the variety now. If you can track the grain, wheat is a global grain crop, right? So it's traded and a lot of it's exported. Our wheats are known for high quality, exported all around the world. So, I don't know whether it's going to end up on your plate, but it's certainly very important for the Australian economy. We eat and consume very little wheat grown here in Australia.
Yeah, we've got several questions that are around the complexities and managing those sort of things. So a couple of them together. Firstly, are there any negatives in speed breeding? And if so, what are they? And related, how do you ensure that speed breeding doesn't introduce undesired properties? So the example given was heat tolerance declines while trying to improve disease resistance.
What was the first question?
The first one was just, I guess, more broadly, are there any negatives in speed breeding?
Negatives? Oh, yeah. Look, surely one of the negatives is the cost. Energy is like all these technologies. Energy is a key factor, and that's why I think there's a real challenge in everything that we do is all about energy. How do we reduce the cost of energy? How do we come up with renewables? How do we fuel all this stuff? Electric cars are booming, So I think, that's certainly one of the major challenges. In terms of the negatives and trade-offs, perhaps, there's always going to be, a risk of not integrating your desired trait if you're not selecting for it. So because these traits are controlled by many, many genes, if we're not selecting for that trait along the way during development of our populations, then we lose the genes because we might be selecting for disease resistance, and we get to the end, we have an almost finished product, a variety, but it doesn't have the heat tolerance. So unfortunately, that means we have to make another cross and go back all the way through the whole breeding pipeline again. And that could be another 10 years or longer until we get to that same stage where we recombine all the genes.
Great. Question, a couple of different questions on the same theme, but boiling down to, why are all the lights in the speed breeding labs purple?
Yeah, well, it's pretty cool. Just to make it cool. No, it's.
You have the disco ball out and the Saturday night.
The plants go crazy at night time. No, well, look, it's because these new LEDs are only emitting the blue and the red wavelengths, and so together it creates this sort of purple haze. Not just because we've got this, purple is our UQ color. And so, look, that's why the normal regular lights that we have appear yellow. They're not producing, or they're producing extra wavelengths. The plant doesn't need all these other wavelengths, and they're reflecting these other wavelengths, and that's why they appear green. As soon as we expose them only to blue and red, they actually look like blackish purple color in a completely closed system. It's really trippy. You got to get those plants out into the daylight and check if they're really green. But they are green when you take them out to the daylight.
Good. You got energy for a few more questions?
Yeah, sure.
We have a lot of questions.
Away. I mean, I'm on the spot here,
So, a question around from Damien. Who owns the intellectual property over the new varieties resultant from speed breeding?
Good question. Well, if we developed materials ourselves, and we were funded by the federal government to generate those materials, or from a body like GRDC that's funded by the farmers, then generally those materials are made available to all the companies equally. And however, we do work closely together with some companies, and that happens under various contracts and arrangements. We want to encourage investment in our crops and future development, and so under those conditions, it depends on the project. But we might have a stake in the IP, we may not, and we collaborate for different reasons. It all depends. But what we're trying to do with the methodology of speed breeding is to spread the word, make it all open access. That's why we made all of our protocols freely available. And I'm very open, and I'm traveling the world and trying to share as much information as I can with my fellow scientists and plant breeders. So, yeah.
A couple of questions from Margaret on Twitter and one of our audience here around, does the nutritional value of speed-bred crops change? And do you check for nutritional value as well as other aspects?
Yeah, so it's a tricky one. The nutritional value is controlled by many, many genes, and those genes actually interact with the environment. And so I guess what we're trying to do in a speed breeding system is not lose those genes. So as long as we have those genes in the plants, when they get to the field, they will produce the seeds that have high nutritional content. Right? In the speed breeding system itself, when we're cycling quickly, we're not too concerned about the quality of the seed. And in fact, the seeds are not very good quality because we're pushing them so hard. Often they're quite shriveled. One of the tricks we use actually to shortcut the generation time even further is we actually harvest the grain green only 2 weeks after they flower, and we dry them down. It's like a fast-track maturing process, and they turn brown, but they're shriveled. then we give them a bit of water, they soak it up, they get fat again, but they don't want to germinate. There's an imbalance of the hormones in the seed that are needed for germination. There's an accumulation of ABA. And so to overcome this biological barrier, we actually just put them in the fridge for a few days and then pull them out and they all germinate. Hey presto, 95% germination. So this has been a real innovation in itself, because before this, people actually had to take them to the lab and rescue the embryos and culture them up. And we can't do that from a plant breeding perspective, because we need to grow hundreds and thousands of plants at a time to play this gene number game that plant breeders are playing to combine all these traits together in a package, a variety for our farmers.
Great. All right, last question. Again, we had a lot of questions tonight, so please do take up the chance to chat more to Lee. The last question is, and I'm guessing maybe from up the back, is what can our school do to help speed breeding? And there were a few questions around, what's next? Is there opportunities for backyard scientists? Is there opportunities to grow crops like this in your backyard? What's next?
Good question. I think there's a lot more science to be done when it comes to speed breeding. There's optimization of these protocols to grow different species. We've only trialled a couple of different crops, but we want to get the system working for other species like rice, sorghum. But this means we've got to do the science. We've got to do the experiments. We've got to test different photo period regimes. How long do we expose the plants for? What temperatures do we use? So I believe that we can speed breed any species, but we've got to do the science and work out what's the fastest way to grow the plants.
Great.
Yeah, and like I think I hinted at the end of my talk, that I think there are opportunities to take these approaches and think out of the box when it comes to indoor farming. Out of the box, indoor farming. But it's again, we have to do the science and work it out. A great example is the tomato plant. The tomato plant is really sensitive to continuous light, so the leaves go all necrotic. But the scientists in the Netherlands, they actually identified a gene which makes the plant tolerate the continuous light. They put the gene into one of their tomato varieties, grew the plants under the speed breeding system, continuous light, and it boosted fruit productivity by 20%. And this is in the Netherlands. They're the world's biggest exporter of tomatoes. So I guess it goes to show that even like a plant like tomato that was sensitive to these conditions, we can find the right genetic variant out there in the wild and use it to boost productivity indoors.
Brilliant. Well, look, that is lots of things to think about, and thank you so much for that. We will have chance for some food and drink and chance to chat to Lee, although if you've got vegetables, you can't have any, you're going to eat those first. Put your hands together for Lee Hickey from UQ.
Excellent.